Okay, so this video is on protein synthesis, and it's actually part one because it's such a big topic. We need two parts. The first part is on transcription, which is how you get from DNA to RNA, and the second part is on translation, where you go from mRNA to protein. So, in terms of this, you have to understand around mRNA and how that works to become an amino acid sequence at the ribosomes, and describe all these different parts and how they work together: DNA, mRNA, tRNA, rRNA, and then look at the genetic code, which we do in the next video. So this is an important part of biology. It's called the central dogma, and it basically says that DNA can be replicated to make more DNA, which we've already looked at, but also DNA is transcribed to make RNA, which is translated to make protein. And this is the connection between the three main macromolecules that we're looking at and the core of this topic, DNA and proteins. Now, it's important to make the distinction that this is not saying that DNA turns into RNA or that RNA turns into protein. What we're talking about here is the flow of information. So DNA encodes or provides the information or codes for an mRNA or an RNA strand, and that an mRNA strand will code for the information to make a polypeptide, which folds into a protein. So polypeptide is just a string of amino acids that have been covalently linked together, and these form a protein. So let's look at that first phase. So this is happening in the nucleus. What we have here is that, just reading this myself, in prokaryotes, of course, they don't have a nucleus, so it doesn't happen in the nucleus. For them, it happens in the nucleoid region, but for eukaryotic cells in the nucleus. But either way, you have an enzyme called RNA polymerase, which will bind to the DNA double helix. We unwind these strands so that the RNA polymerase can get access to this, add free nucleotides, a little bit like DNA polymerase making a double helix, but instead of making two copies here, what we're doing is we're just making a single-stranded RNA using this DNA as a template. And again, the role of the polymerase here is to attach free nucleotides together using base pairing rules in order to make a new macromolecule, in this case RNA, a new polymer. As we saw in the genes video, and you can connect to that as well, this RNA will have some introns that need to be spliced out, and also this mRNA, or this RNA might be an mRNA, which can become a messenger for making protein, or it might have other roles. So as we see down here, there's mRNA, tRNA, and rRNA, and we'll look at that in a sec. But this was really our first opportunity to look at DNA and RNA and the differences between them. Here they're just coloured blue and red, but really there are quite a few differences that we need to look at. So first of all, DNA and RNA are very similar, and they're both made up from 4 nucleotides that are joined together along a sugar phosphate backbone. But the key difference here is that the sugar phosphate backbone, the sugar in that is ribose in RNA and deoxyribose in DNA. The other thing here is that with the 4 nucleotides, we have A's, G's, C and T. On this side we have A, G, C and U. So uracil replaces thymine. And so uracil has the same base pairing. It base pairs with adenine. So A and U go together, C and G go together. Next, quite obviously, DNA is double-stranded and RNA is single-stranded, although it can wind round and bind to itself. And the last one is that where there is a nucleus, the DNA will stay in the nucleus, whereas RNA is made in the nucleus and then travels to the cytoplasm in eukaryotes. So over here, just a little diagram. We don't need to know heaps about this at this stage, but just be aware that DNA can make 3 different types of RNA. mRNA, which is used as that messenger, like we said. tRNA, which we look at a little bit in the next video, which combines with amino acids and brings those to the ribosome. But also there's ribosomal RNA, rRNA, which teams up with some proteins to make ribosomes. So in order to do translation, which is our next video, we need some tRNAs, we need some ribosomal RNAs working with some proteins, and we need the messenger itself, the messenger RNA. And those will make polypeptides, which fold together to make proteins. We'll talk a lot about that in the next video. Here's some additional resources that you may want to look at, but probably the main thing to do is go to the next video on protein synthesis translation and follow through the story.